Welcome to PSB 2021. It's not the PSB that any of us were hoping for, but in these dark COVID times, it's the best we can do to bring you the good science that we can from wherever you happen to be. We want to start by acknowledging the terrible cost that COVID has taken from all of us, and look forward, we hope, to recovery in 2021, appreciating the science that brought us extremely rapid and apparently effective vaccines, and our deeper understanding of how this virus works. It's also been a demonstration of the power of open science, which we will continue to give our PSB Parasite and Symbiont Awards, which will be a little later in the program. Our format this year consists mostly of 10-minute pre-recorded talks, followed by 5 minutes of live Q&A, and each session will have an hour of live open discussion. We also have 2 virtual poster sessions where poster presenters will be in different discussion rooms so you can interact with each poster presenter. The meeting concludes on Thursday with a series of live workshops. You can follow us on Twitter. We are at PACSYMBIOCOMP, or you can go to our website at psb.stanford.edu, which will stream the Twitter feed. We'll be posting updates there in case there are any issues. And our hashtag is #PSB21 this year. So please tweet what you like from what you see. And I want to say that while I enjoy being here by myself, I really miss all of you. And I would like to see you all here next year at PSB 2022. We are now accepting session and workshop proposals. They are due by Monday, February 15th. The call for proposals is at psb.stanford.edu. Or you can email me, Larry Hunter, at cuanschutz.edu, if you have any questions about preparing proposals. And with that, I'm going to turn it over to the scientists, and we will hear some great science. Stay tuned for some really interesting PSB from wherever you are.
Hi, everyone. First off, I'm just going to make sure, can you hear me? That is the phrase of 2020. So
we can hear you great, Roxana.
Perfect. Awesome. All right. Hi, my name is Roxana Donishew, and on behalf of my co-chairs, I am excited to introduce the opening session for virtual PSB 2021. Our session today covers computational challenges and artificial intelligence in precision medicine. So I want to just cut through the buzzwords to talk about what this really means. I think this is an incredibly exciting time for bioinformatics in medicine because of the large amount of data that we have access to. And as a physician, I know in medicine, we already provide tailored care to our patients. However, with precision medicine, we are leveraging additional data, whether that's from genomics or other omics modalities, clinical imaging, wearables, electronic health records, to try to further improve personalized diagnosis and treatment. And that is what we call precision medicine. And of course, there are many more things that go into it than just on this slide, but these are some of the modalities and data sources that we take in when we're talking about precision medicine. This deluge of multimodal, multidimensional data, however, is more than any one human can interpret. At least it's more than I can interpret as a physician. And then, as many of you know firsthand, there are scores of challenges for dealing with this data, from medical data access issues and data structure to formatting to regulatory hurdles. Furthermore, given the bias in our society, we always have to ask ourselves, does our data leave certain groups out? And how does this exclusion perpetuate bias in our algorithms? And so we're dealing with all these sorts of different challenges as we're trying to massage these large amounts of data. It's these challenges that our computational tools hope to address. And by addressing these challenges, these computational tools can kind of help us parse healthcare data and gain new insights that can hopefully improve patient care. These days, many of our methods for parsing and interpreting complex medical data involve artificial intelligence. Now, I know it's common to see artificial intelligence, machine learning, and deep learning used interchangeably. But when we talk about artificial intelligence, we're thinking about techniques that help computers mimic human behavior. For example, one example of this kind of human behavior might be identifying a concerning lesion on a chest x-ray, the way that a radiologist does. Machine learning represents a subset of artificial intelligence, or AI, in which we use statistical methods to help machines improve with experience. And deep learning is a subset of machine learning that uses multi-layered neural networks to perform often complex tasks like image recognition. Today, we're going to see the way that some of these tools have been applied to multimodal data sets to gain new insights. So we'll have a series of talks that deal with omics data. This data helps us gain insight into the role of the genome and genomic regulation in human health. We will learn of new methods for studying the impact of rare genetic variation on genome regulation. We'll learn about the role of genetic variation in influencing drug phenotypes or pharmacogenomics in large data sets. And we'll talk about methods for identifying methylation adjusted eqtls in datasets representing historically excluded populations. We also have a series of talks on dealing with image-based data. With the COVID-19 pandemic, telemedicine has rapidly increased, introducing new challenges with photo quality. Machine learning tools for radiology and for the interpretation of chest x-rays have been an area of great excitement. But today we'll take a step back and learn about the potential biases introduced when diagnostic machine learning models are trained on publicly available chest x-ray data sets. So while we're not on the Big Island, and we won't have the opportunity to sneak away from the conference to see a volcano or snorkel with sea turtles or go to the beach, I hope you will still enjoy the intellectual stimulation that PSB in our session provides. So we will go ahead and begin to play the videos. Feel free to put any questions into the chat box, which we will be monitoring, and there will be a 5-minute Q&A with available authors afterwards. One more logistical thing, at the end of all this, we're going to have a 1-hour discussion. And so if authors can please stay on board for that, where we can ask additional questions and stimulate, conversation around some of the topics that we talked about today. Thank you again, everyone, for attending, and I hope you are staying safe.
Thank you, Roxana, for the introduction. Now I will start playing video. So we will have the first talk by Guanlan Dong on eqtl analysis using region-based aggregation of rare genomic variants. And as Roxana was mentioning, please ask questions during the talk in the Q&A box in your Zoom interface. And we'll have a Q&A session just after the presentation. Let me share the screen now. Okay, so it should be running in a second. If you don't see the screen, let me know.
Hello, everyone. Today, I'd like to introduce you to our new tool called eqtl, which performs eqtl analysis using region-based aggregation of rare genomic variants. First off, eqtl stands for expression quantitative trait loci, which are defined to be genetic variants associated with variations in gene expression. With the advancement of sequencing technologies, there's been more large cohorts with concurrently available DNA and RNA-seq data, which has provided a lot of opportunities for association-based eqtl identifications. However, for rare variants, the underlying power of the statistical testing is often limited due to the small carrier sample size. And one way to address this challenge is by increasing carrier sample size through variant aggregation. There's been several methods developed in disease association studies where they test for group-wise variant association with disease status. Shown here is one of the methods developed by Li and Leo, where they demonstrated using simulation data that variant aggregation indeed yielded higher power than single marker test. So what we have right now are existing variant aggregation methods which focus on associations between grouped variants and complex traits and disease status. And on the other side, we have existing eqtl tools which focus on associations between single variants and gene expression. So that's why we developed eqtl to bridge this gap between existing variant aggregation methods and eqtl tools by testing for associations between grouped variants and gene expression. eqtl is specifically designed to accommodate standard file formats generated from sequencing data, such as VCF files and map files. So first, we wanted to see whether variant aggregation works in the eqtl context. So we performed a simulation analysis using 10,000 independent simulations for each sample size. Shown here is the power plot, where you can see that group variants, shown in green, consistently yielded higher power than single variance testing, shown in blue. Also note that to reach a power higher than 95%, group variants only required 30 to 40% of the sample size needed by single variant testing. Let me just walk you through the workflow of eqtl. It requires 3 input files: expression file, a genotype file, and the region file. So both expression file and genotype files are standard file formats where you can just directly input the sequencing data into eqtl. While region file is completely customized by the user and provides a lot of flexibility in study design, where the user can specify genomic region and genes to be tested against each region. eqtl can also incorporate an optional covariate file, which can contain information such as clinical factors and population substructures to further refine the association. So first, eqtl would use genotype file and the region file to map variants to regions using an interval tree algorithm. And once the variants are grouped together, it would then partition expression based on such grouping. Then together with covariates, if provided, eqtl would conduct an association test for each gene using a multiple linear regression model, shown here. So for sample I, E stands for the gene expression, G stands for the collapsed regional genotype, and C stands for a vector of covariates. Finally, eqtl would perform multiple testing correction using the Benjamin-Hochberg method and output a summary statistics file which includes all the regression coefficients and raw p-values, also the adjusted p-values for all the components in the model. So to demonstrate that eqtl could identify eqtls in real data set, we applied it on germline TCGA data. We tested the hypothesis that rare germline truncations in cancer susceptibility genes are associated with their cis expression in breast cancer patients. Shown here is the QQ plot of the results, with blue indicating genes with adjusted p-values smaller than 0.05. And as expected, BRCA1 is one of the top-ranked genes identified by AEQTL, and other top hits have also been reported by previous studies with implications for cancer. Aside from germline variants, we also applied AEQTL on somatic mutations across 32 TCGA cancers. Out of 300,000 truncations and 2 million missense mutations, AEQTL identified thousands of significant variants, which also have a relatively large overlap with this list of likely driver genes reported by Bailey et al. And many top-ranked genes were not previously identified drivers, which could represent new candidates of the long-tail driver genes with lesser mutations reported by multiple recent studies as well. Next, we interrogated this 299 likely driver genes. Shown here are QQ plots with gene cancer pairs ranked by their adjusted p-values, which can also help prioritize those likely driver genes for other studies. Stemming from our likely driver gene analysis, cancer drivers can be classified into oncogenes and tumor suppressor genes, or TSGs, depending on their mutated cancer type and pathway context. However, most existing classifying methods leveraged mutation data that lack considerations of their downstream consequences. So we wanted to know whether we can use eqtl patterns to predict driver gene labels. And interestingly, we found that the genotype regression coefficients of somatic truncations showed a strong association with their predicted classifications. From this violin plot, you can see that genes predicted to be oncogenes had larger positive genotype coefficients, while genes predicted to be the tumor suppressor genes had larger negative genotype coefficients. So to further verify our finding, we also performed a receiver operating characteristic analysis and yielded an area under the curve of 86.3%, indeed suggesting the potential of using somatic truncation eqtl patterns to distinguish between oncogenes and tumor suppressor genes. To quickly wrap it up, we have developed a new tool called eqtl, which increases the power of rare variant eqtl identification by aggregating variants in defined genomic regions. We applied it on TCGA data to demonstrate its capacity of identifying both germline and somatic eqtls. And it also revealed a potential new method of using somatic truncation eqtl patterns to classify driver genes. eqtl is a command line based tool implemented in Python, and it's easy to use and customize because there's no need for excessive pre-processing on two of the input files, genotype and expression files, while the region file allows great flexibility for study design. So here I mainly talked about the applications on cancer data, but with increasingly available cohorts of matched whole genome and transcriptome data, we expect that eqtl can be adapted to a much broader range of eqtl studies. Last but not least, I would like to thank the Cancer Genome Atlas and its participating patients and families for generously contributing the data. I would like to thank all my co-authors for providing their insights and edits on the manuscript, and especially my mentor Kwan for mentoring me throughout this entire project. Here is the GitHub page of our tool. Please feel free to give it a try. And if you have any comments or questions, you can also reach me at this email address. With that, I'd like to thank you for your attention, and the real me there would be happy to take questions now. Thank you.
Thank you very much, Guanlan, for that presentation. I'm Martha Bullock. I'm going to be moderating the QA. We have a few questions already in a QA box. So the first one is, how does AEQTL deal with chromosomal rearrangements that often happen in cancer genomes?
So right now, eqtl mainly is accommodating SNPs and SNVs and indels. So unless chromosomal rearrangements are annotated as deletions or insertions, it wouldn't be considered by eqtl.
Is that something that could be incorporated in the future?
I think so. I believe so. Definitely.
Okay. Next, how are the multi-SNP genotypes encoded? Maybe provide a 2-SNP genotype example. And then a follow-up question is also, did your simulated power calculations assume that all included variants were associated with the phenotype? If so, does the power decrease as more non-causal variants are included in the multi-SNP genotypes?
So for the multi-SNP genotype scenario, we're not taking into account of multiple alternative alleles, if that's what they're asking. So basically, as long as there's one alternative allele that's different from the reference allele, like that would be sort of grouped together as the one situation. And in terms of the simulated power calculation, we simulated 10 variants per simulation, where 5 of them were sort of like effective, basically meaning that they are associated with the phenotype. So yeah.
Okay. And then William Bush asks, for the AEQTL talk, I was just wondering if you examined any of your truncation variants to see if they trigger nonsense-mediated decay.
So that would, in our manuscript, we actually sort of discussed the nonsense-mediated decay, especially considering the sort of truncation patterns we discovered in TSGs and oncogenes. That was one of our most favored guess, but we didn't really have time to look further into that. But that's something that we are hoping to do for sure.
Any other questions? All right, thank you very much, Guanlan.
Thank you so much.
Great, thank you very much. Let me share the second video then. The second presentation is by Kailas Voderhari and Roxana Danesh-Ju. I will share the screen and please ask questions as previously in the Q&A box.
Hi, my name is Roxana Donishu. I'm a practicing dermatologist and a postdoctoral scholar in biomedical data science at Stanford, and I'm here with my colleague.
I'm Kailas Vajrahalli. I'm a second-year PhD student at Stanford.
And we're here to talk about True Image, a machine learning algorithm to improve the quality of telehealth photos. When the COVID-19 pandemic hit, hospital systems were overwhelmed as they tried to move things to telemedicine. In fact, there was a 10,000x increase in digital visits. Dermatology was part of this increase. Suddenly, instead of seeing patients in person, I was seeing 25 to 30 patients a day, all through televisits. However, because the video quality was not very good, I was asking patients to send photos in ahead of time because good quality images are vital to practicing good teledermatology. What my colleagues and I quickly realized is that even with instructions, it's difficult to take good photos. In fact, when we talked to our colleagues and took an informal poll, we found that greater than 20% of the images that were sent in were poor quality. These images had to be reviewed manually before each visit, and then patients had to be called and told to please send in a new image, and sometimes we would discuss with them what they could do to take a better photo. If the patient didn't send their photo in until right before their telederm visit, what ended up happening is the entire visit would be spent talking about the photo quality instead of focusing on the clinical need. This ended up wasting significant amount of time for both the clinicians and the patient. Here are some examples of the photos that we got that were not good quality. The first image is too blurry for us to see what's going on. The second image and the third image both have lighting issues. Other things we noticed were issues with cropping, so being too zoomed in or zoomed out. These things made it difficult to assess what was going on. That's why we created the True Image algorithm. The same way you're able to take a good photo of your check for online deposit, we wanted to create an algorithm that would allow us to take good quality photos for teledermatology.
Now I'm going to describe what the true image workflow would look like to a patient. After the patient takes an image, the true image algorithm will assess the image and determine whether or not it's good quality. In this case, the image the patient took has poor lighting. So the true image algorithm will tell the patient to move to an area with brighter lighting. The patient will move to a brighter lighting area and retake the image. Now the image is good quality, and so the true image algorithm will automatically submit it to the clinician. This makes the process much simpler for a patient, as one, they don't have to worry about how to take good images, and two, for the clinician, it makes it much simpler as they are guaranteed to get good quality images. Now I'm going to briefly describe what our algorithm looks like. I'm only going to talk about it at a high level. For full details, please see our paper. First, we start with an image. This image is then segmented into background pixels, skin pixels, and lesion pixels. This is a very important first step in our algorithm, as we don't care about the quality of the background pixels, but we care very much about the quality of the lesion pixels. Using this segmentation, we generate three types of features: blur features, lighting features, and zoom crop features. Blur features are used to distinguish blurry from non-blurry images, lighting features to distinguish images with poor and bad quality light and good quality lighting, and zoom crop features are used to distinguish between images that don't have these issues and images that do have these issues. After we generate these features, we concatenate them together and we train 4 different logistic classifiers. The first logistic classifier is used to classify whether an image is good or bad quality at a high level. The remaining 3 classifiers are used to label whether the image has issues with blur, issues with lighting, or issues with zooming or cropping. The workflow, the way the algorithm works is in 2 steps then. The first step is to use the first classifier to label the image. If that label says the image is bad, then we use the remaining 3 classifiers to determine why the image is bad. In this case, this image actually is good quality, and the first classifier will return that the image is good quality. So we get the label that the image is good. And in the workflow I previously described, this would then allow the image to be sent directly to the clinician. This image, on the other hand, is bad quality. So the first classifier will return it is a bad quality image. And of the remaining 3 classifiers, the one that is responsible for lighting will return that there is a lighting issue. Similarly, this one will have a blur issue, and this one has zoom and crop issues. Here I'm showing the current results for our first good-bad classifier. This is an AUC plot, and you can see that we have reasonable performance. If we decide to put a threshold in our algorithm at this point, we can reject 80% of poor quality images while retaining 60% of good quality images. Now, 60% of good quality images may not sound that good, but in fact, it's actually not that bad at all. And the reason is it's very easy for patients to take multiple photos. In fact, the patient can take multiple photos in quick succession, and if any one of those are good quality, then they'll make it through, because you only need 1 image to send. Our blur classifier actually has much better performance. And if we set a threshold here, we can reject 90% of good quality of bad quality photos while retaining over 80% of good quality photos. And I want to emphasize this result because in practice, we've seen that blur is the most common issue out of the images patients send in. So having a good blur classifier is actually very helpful. Here I'm showing our results for our lighting and zoom crop classifiers. As you can see, the results are not as great. This is primarily because we don't have as much data for issues with bad lighting and issues with zoom and crop issues. This is something we are actively working on, and I'll discuss in a bit. So now I want to discuss two future directions that we are currently pursuing. The first is to get additional data. And as I just mentioned, our focus is to get additional data with lighting issues and with zoom and crop issues. Increasing our data set size in general, we can use that to improve our algorithm. And with images with the poor lighting and the zoom and crop issues, we can focus on improving our classifiers for those issues. In parallel, we are also working on deploying our algorithm in a clinical setting. This will allow us to assess how patients can use our algorithm and also how clinicians can use our algorithm. So we can look at ways of improving it and look at where it fails and where it's especially useful. Lastly, I want to acknowledge our collaborators, Dr. Robert Ternovoa, Dr. Albert Chu, Dr. Justin Ko, and Dr. James So. Thank you.
Thank you very much for that presentation. So we have a couple questions in the QA. First question is by Yiran Liu, who asks, what skin tones were included in training and/or test?
Yeah, so we included a variety of skin tones, and this is a project we're still working on, and we have a whole variety of skin tones. So people with light skin, people with dark skin, etc. And this is something that is very important because in practice, we will get people with a variety of skin tones. So we need to make sure that our algorithm actually does work across different people.
We were really intentional about that, actually. Yes. And in our collection too, we are still collecting images, and we're being very intentional about skin tone diversity.
Next, we have an anonymous attendee who asks, did you investigate whether the algorithm performs equally well on different ethnicities or age groups?
Yeah, so that's something we definitely are interested in, but we have not investigated yet because at the time, we didn't have a lot of data, but it is something that we are collecting data on. So yes.
Okay. Samuel Sledjewski asks, how did you obtain labeled data for good, bad images, blurry, not blurry, etc.?
Yeah, so for this, so in the stuff I presented, that was just us labeling. We now have set up a flow where we are asking residents to labels for us. So we are getting labels from actual clinicians who are evaluating it. And this is important because for different types of diseases, an image may be considered blurry for one, but not for another. So it actually is important to get clinical feedback on what is a good quality or not good quality image.
I'm actually curious, were you able to check whether your performance was better for certain diseases than others?
No, because not enough data really to validate that.
Okay. William Bush asks, how do you account for the variability in camera quality? Is there a minimum requirement?
For this, we haven't really done this analysis yet because the data we have doesn't necessarily have what phone the image was taken on. So that is an issue. So currently there is no minimum requirement, but there definitely is variability in camera data. So it potentially is an issue we need to consider later on.
The images we have are actual real patient images. So they're actually coming from various cameras. And in practice in telemedicine in general, there is no, like, patients are not told you have to take the photo by a certain camera. So we're using real world data. And so that's kind of representing the full spectrum of what you might see in terms of like smartphone cameras.
Do you collect information on what kind of camera they used?
No, we don't, because it's retrospective. So it's from past telemedicine visits. However, we are actually working on a prospective trial right now where we may look at whether or not that has an effect.
Great. Next, we have a few different anonymous attendees who ask, let's see, starting with, was the algorithm better at identifying certain types of lesions than others? I think that is like the question I'd asked.
Yeah.
Is there a minimum resolution needed for the algorithm to properly read the image?
So there isn't a minimum resolution, but if it's too low, it'll be classified as blurry. So Generally, it's larger than 512 by, let's say, 256 or so.
Okay. Another anonymous attendee question is, might your algorithm classify a normal photo of clear skin with no issues as blurred?
So it depends, but I would say no, because clear skin is not really blurry looking. There is variation in the skin tone in just a normal image. So I would say no. The answer should be no.
Okay. Peter Washington asks, could the data set be augmented with blurry images artificially?
Yes, and we do that. So you can check out our paper for how we do that.
Okay. William Sutherland asks, is there a difference in success rate based on skin tone? I think we talked about that.
Yeah, we talked about that a bit, and it's kind of like we're trying to validate this and make sure this is there is no difference in success rate.
Okay. Stephen Hart asks, have you considered extracting metadata from the image itself?
What kind of metadata, I would ask. So we haven't really done that yet. There isn't a whole lot of metadata we can extract because we have limited information from the images, but yeah.
Yeah. Sometimes the images are we're not actually getting the original file of the image. We're actually getting the image as it was like pulled from the EMR system.
Okay, we have an anonymous attendee asking, what considerations did you have for picking your thresholds in your ROC curve when applying to the clinic?
So what in the slides I showed, it was somewhat arbitrary. When we actually want to deploy this, we'd have to talk to clinicians on what they need, and we have to validate this in clinical trials to see what patients need, to see what makes sense. So it's not something that's fixed. What I showed was a little bit arbitrary, just to show an example of what it could look like.
Okay. Marix Faboda asks, can the fact that there are no camera resolution requirements mean that images from some cameras may never be accepted by the algorithm, resulting in patient frustration?
I mean, that would be possible. In that case, that means the patient can't actually take a good image on their camera, which is an issue in and of itself. I think when we actually want to deploy this, we would have ways for the patient to actually submit an image around the algorithm, just in case. But yeah, this would be more of an issue of the patient can't actually take good photos at all with their phone then.
Stephen Hart, who had asked the question before about metadata, wrote back clarifying that he's referring to compression type and image content.
Yeah. So like Roxana said, we generally don't have access to this information. So.
All right, and then we've time for one final question from Morris Aguilar. Can image adjustments like increase in contrast, sharpness, or exposure adjustment improve the model's performance?
Yeah, so we actually tried that. I don't think it's in the paper, but we did try this. It did not seem to help much.
Perfect.
All right.
Thank you.
Thank you very much.
So since we are still ahead of schedule, I have one short question. So I was wondering, why did you end up decomposing the algorithm to this blurring lighting instead of using the end-to-end approach? Like, what's the end-to-end deep learning approach?
Yeah. So I mean, end-to-end deep learning would be great. There's two reasons. The first reason is we have limited data. At least at the time of this, we had limited data. So a deep learning approach wouldn't have made sense. The second reason, which is a bit more general, is we do want to deploy this type of algorithm on a phone. And as computationally efficient as possible is better. So deep learning is usually more computationally inefficient. So that's why we didn't go for a deep learning approach. But it definitely is a possible option. Makes
sense. Yeah, thank you. Sounds great. So let me continue sharing videos. Now we have, just a second. And the next presentation is by Lele Sayed Kalantari on fairness gap in deep chest X-ray classifiers.
Hi, my name is Lale Sayed Kalantari, and I'm here to present our work, exclusion, fairness gap in deep chest X-ray classifiers. Maya was a student. Unfortunately, medical condition which affects her ability to function. Initial assessment didn't show any disease for her, therefore she sent home without further investigation. Researchers have found the evidence of racial, sex, and socioeconomic bias in healthcare system. Race and sex bias exists in disease underdiagnosis and medical risk score analysis tools. The quality of care delivered to a patient even varies by their insurance type. Female receive a longer time to diagnose than male, though they may show the same medical condition. This can potentially deprive minorities of the ability to receive quality health care. Chest X-ray imaging is an important screening tool for several life-threatening diseases. However, the shortage of the radiologists put the patient life at risk. Medical image pathology classifiers are one potential solution for the shortage of radiologists. In these classifiers, a medical image is given to a network, and the potential of having a number of diseases is the output of the network. These classifiers has shown good performance, especially recently in diagnostic and comparable to the radiologist level performance. This makes them a good case for deployment of this model. During the training and evaluation of these models, usually we train them and evaluate their performance on overall population. And we do not check how are they performing in different subgroup of the patient within the data set. For example, in different race, sex, age, or insurance type. We want to see if all patients have equality of opportunity in correct disease diagnosis regardless of their sex, age, race, and insurance type. To answer this question, we measure the difference in correct diagnosis, in other words, true positive rate, among different protected attributes. We define the true positive disparities as the difference in true positive rate between the subgroups. And we evaluate it over our data set over subgroup of sex, age, race, and insurance type. We did this study on 4 data sets, Mimic CXR, Check Spread, and Chest X-ray 8, and also we on a multi-source data set which aggregate all 3 of them together on 8 shared labels that they have. All these 4 data sets are not sex skewed. Patient older than 80 years old and patient younger than 20 years old are minorities. Mimic CXR, in addition to the sex and age, also has the race and insurance type of the patient. For race, we can see that white patients are the majorities. However, patients with Medicaid insurance are the minorities in insurance type. First of all, we show that our classifiers is good, and it is having a good AUC average across all patients. Afterward, we calculate the true positive rate disparities per subgroup per disease. For instance, in this image, you can see the true positive disparities of patients with respect to their insurance type over all 14 diseases of Mimic CXR dataset. If we have a 0 disparities, then we can say that our networks is fair. However, if a subgroups has a positive disparities, we call them, they are favorable subgroup, which means that the networks behave in favor of that specific subgroup. Also, if a subgroup receive negative disparities, it means that the classifiers behave not in favor of that specific subgroup. For instance, you can see here poor other. You can see that patient with Medicare insurance has the most positive disparities, and patient with Medicaid insurance has the least disparities, which is negative. And also you can see that the scatter plot of Medicaid insurance is smaller compared to the other insurance type. The size of this scatter plot per disease is varied by the number of patients, by the percentage of the patient that are belong to this specific disease. You can see that here we show the gap between the most and least favorable subgroup, and we average these gaps over all 14 labels in Mimic CXR dataset, and it has been shown here. Ideally, we like to have zero gap. Support device has the lowest gap, where plural other has the largest gap. Patients with Medicaid insurance are commonly receiving negative disparities compared to other subgroups, so they are unfavorable subgroups. On the other hand, the patient with other insurance are commonly receiving positive disparities, and they are favorable subgroup. In this table, you can see our results over all attributes and for all data set. For sex and age, where we have this for all data sets, all 4 data sets, you can see a multi-source data has a lower average true positive disparities compared to all single source data set, which is showing that if you use a multi-source data, it may also help you a little bit to have lower disparities. But these disparities still exist, and using a multi-source data set do not remove them completely. Here you can see that female, patient younger than 20-year-old, Hispanic, and patient with Medicaid insurance are usually among the unfavorable subgroup, which means that all these classifiers are often biased against these specific subgroups. One question is that, are these disparities proportional to the membership? In other words, is the reason that we have disparities against Medicaid insurance is because we have lower number of patients with Medicaid insurance for a specific disease compared to other disease that on other subgroup within the same disease. To answer this question, we calculate the Pearson correlation with Benfroni correction between true positive rate disparities and disease membership. We find that in majority of hypothesis tests that we did, the correlationship is positive, but only in 6 out of 33 of them, this correlation is statistically significant. Our study shows that there is a significant bias in all 4 data sets that we have studied. The chest X-ray medical image classifier do not correctly diagnose patients who are female, less than 20 years old, Hispanic, or the patient with Medicaid insurance who are usually low-income patients. These patients are not receiving correct diagnostic at the same rate to the overall population. Also, our study shows that these biases are not often statistically significant correlated with disease membership. Therefore, if you just use a balanced data set, you will not be able to remove your biases. The classifier trained on a multi-source data has lower average biases compared to single-source data set. Therefore, it's good to use multi-source data set. However, it will not remove all the biases. AI help us to show that these biases exist, and it may help us in the future to mitigate these biases. At the end, we encourage all clinical decision makers to carefully audit for fairness prior to deployment of these models. Thanks for your attention.
Thank you very much, Lala. So maybe I'll start with a question. I was wondering if you have thoughts on how classifiers could be improved, or how the training data could be improved to alleviate some of these current biases.
Sorry, I just had a technical problem. I cannot like open my camera. Is it something that I should do?
You turn your video on?
Yeah, I turn it. It doesn't let me. It says the co-host has asked you to start your. Oh. Okay. Okay. Yeah.
We can see you.
Yeah. Okay. So, what was the question?
I was wondering whether you have suggestions for how either the training data or the classifier could be improved to try to alleviate some of these biases.
I think the training data, for example, we show that if we are using multi-source data, it is improving a little bit. So for sure, if we have more data, that would help us to have a better results. If we have more balanced data, it would help us probably. But what we find out is that like not having the balanced data set is not the only reason for these biases. So it will help us to improve things, but not definitely like completely removing all the biases. Yeah, so I think it's still like playing with the training data would be an important path probably to try to mitigate these biases.
Is there a challenge in getting sufficient training data for those groups?
Usually, like in medical imaging, it's challenging to have access to large medical data set. But in particular, in chest X-ray area, we have access to very large available data set, which is very good compared to other fields in medical imaging. But in general, like among the 3 largest available public data set that we have, only Mini-6R provide the insurance type or race of the patient information, which was very good and which was very informative in our study. And other data set didn't reveal this kind of information. I think if like they kind of put this kind of information in the data set, that would help a lot us to see what's going on in other data set. And if, for example, a data set is doing better, what's specific about that data set that make it better compared to other ones.
Okay. William Bush asks, could some of the noted misdiagnoses be due to machines used, i.e. low-income patients may be seen at hospitals with lower resolution images? Could that affect the classifier more than we realize?
That potentially could be the reason. Yeah, there are, we usually, like we cannot ping something and say that's exactly the reason because it's really kind of hidden and there are lots of factors. If we want to make a very clear answer, we have to do kind of causality study and see, for example, if the brightness of the images are different on those patients compared to the other patient. But that's a good start point to look at this kind of things. Yeah.
Do we have any other questions?
I have a question, if you don't mind, that we're still ahead of schedule. So I was wondering that those algorithms tend to just carry the biases from the data set to the algorithm. And one possible explanation could be that the noise in diagnostics for certain populations is bigger, and the problem of predicting for those populations is bigger. So maybe there was some bias in the data set itself, or like there was a lot of variability for certain subgroups in the original data set that then the classifier actually cannot predict well on those subgroups because on the data we have noise and there was some bias in doctors and so on. Like, is that a possible interpretation that that the problem is not the algorithm but the data itself?
That exactly the same thing. Right? Like those are all potential reasons for that, and yeah, that could be one potential. But like all of those, like we have to look to them more carefully to see like if they are the real cause of this bias. Yeah.
All right, we have one more question from an attendee. It's more of a comment. Bonferroni corrections are the strictest possible correction. So 6 out of 30.
Yes, my computer is going to run out of the battery. Let me do that. Okay, sorry about that.
The comment is that Bonferroni corrections are the strictest possible correction. And have you tried a more powerful method such as home Bonferroni? It's important if sample size is the issue.
We haven't tried that, but I would be happy like if I get more information and I test that also on the study.
All right. Thank you very much, Lali.
Thank you.
Thank you. me share the next video. So the next talk is by Greg McInnes. I'll enter the full screen, and here we go.
Hi, my name is Greg McInnes. I'm a PhD student at Stanford University, and I work in Russ Altman's lab. Today, we're talking about drug response pharmacogenetics for 200,000 UK Biobank participants. So pharmacogenetics is the study of how genes influence drug response. So when you take a drug, it interacts with proteins all throughout your body, whether it's being metabolized in your liver or transported in and out of cells or ultimately hitting the drug target. All of these interactions are with proteins. And variations in the genes that encode the proteins can lead to differential response among individuals. So you could imagine you have some metabolizing enzyme in your liver, and if you have an individual who doesn't metabolize a drug very well, they may end up with more of it circulating throughout their system, which could lead to drug toxicity and adverse events. You could also imagine individuals who metabolize a drug very rapidly and thus break it down and excrete it much more quickly than most people. And for these people, you might not have any therapeutic benefit. And ultimately, the goal of pharmacogenetics is to better understand these differences in how people respond to drugs and use this to improve the prescribing practice. And pharmacogenomics really affects everyone. More than 99% of people have an atypical response allele for at least one drug. And on average, people have an atypical response allele to 10 drugs. And these aren't just some abstract drugs out there that you'll likely never take. These are common drugs that you may in fact likely take throughout your lifetime. These include antidepressants, statins, proton pump inhibitors, things that you or your loved ones will likely be on at some point. Historically, pharmacogenomic studies have been difficult to conduct, partially due to small sample sizes. You can't just go around giving people a bunch of drugs and kind of seeing what happens and seeing if that's associated with their genetics. You have to conduct these trials very carefully. It's typically much easier to do disease studies because you either have the disease or you don't. It's difficult to do retrospective studies because you can't often define clear clinical endpoints for pharmacogenomics. However, with the advent of very large biobanks that are linked with clinical data, we have an opportunity to uncover some of these associations. And to do this, we can use the natural prescribing patterns to uncover these associations. So by this, I mean just when you go to a doctor, maybe you get started on some drug, hopefully it's the right dose and it goes well, but maybe after a month they ask how you're doing and they may need to increase or decrease your dose. And so we can look for these changes through overall patients throughout time to find where they end up through this prescribing practice and see if there's an association with genetics. And using this procedure, we have the potential to support existing pharmacogenetic associations as well as hopefully discover new drug-gene relationships. So in this study, we used data from the UK Biobank to do this. We used genetic and primary care data from 200,000 participants. We called pharmacogenomic star alleles using a tool we developed called PGXPOP. And what this gives us is the pharmacogenomic phenotypes for several genes. For this study, we used 9 genes, many metabolizing enzymes such as CYP2C19, which I'm showing here. CYP2C19 is responsible for metabolizing many drugs, including antidepressants. And we have a range of different phenotypes that we can expect in individuals. So some people are normal metabolizers, others are rapid or ultra rapid metabolizers, some are intermediate or poor metabolizers. And what we would expect to see, there's a big difference in drug response for people who are poor metabolizers and ultra rapid metabolizers and kind of everywhere in between. And these are the phenotypes that I'll be using throughout the study here. The phenotype data is again from these same 200,000 individuals, and this typically goes back about 30 years of general practitioner visits to the doctor. So these are, we have every diagnosis they've ever had from the GP, as well as every drug they've been given, and an estimate of the dose they're given. We don't know if they in fact took it, we just know that they were prescribed the drug, and we kind of hope that they took it. So we conducted 2 analyses for this study. The first analysis was we tried to correlate pharmacogenomic phenotypes with maintenance dose. So basically just, do you end up on a higher or lower dose of a drug, and is this associated with genetics? The other analysis is something we called a differential drug response. So what this means is we were just looking for any difference in diagnosis patterns in the year following a first exposure to a drug. Basically, are you more or less likely to get some side effects if you have a certain pharmacogenetic phenotype? So the first one was the dose association. To set this up, we had to first calculate the maintenance dose, and this isn't in the record. This is one of those problems with the clear pharmacogenomic endpoints. So to do this, we calculated the average prescribed daily dose of the last 5 prescriptions. And on the right here, we can see this distribution of warfarin prescribed daily dose among all patients in the data. And then we performed 2 types of statistical tests to correlate pharmacogenomics with the dosage. We used the John-Kearse-Terpstra test, which is sort of a ranked relationship among the pharmacogenomic phenotypes, and Kruskal-Wallis, which looks for, is there any phenotype that is different from the others? And we did this among 200 drugs, or 9 genes, but we only looked in drug gene pairs that are known to interact. So this is our first set of results here. So what I'm showing in this table here is drug gene pairs with CPIC guidelines. So that's the Clinical Pharmacology Implementation Consortium. These are drug gene pairs that there's a lot of evidence for, so much for that CPIC has written guidelines to instruct doctors how to prescribe these drugs based on genotypes for these genes. And we get an old friend as our #1 hit, warfarin and CYP2C9. This is one of the most famous pharmacogenomic associations with an extremely low p-value. And you can see here that there's this clear kind of linear relationship from poor metabolizers to intermediate metabolizers to normal metabolizers being on increasing dose. We see a similar pattern for phenytoin and then a number of other drug gene pairs. We also looked in drug gene pairs for which there is some evidence in the literature that there is an interaction, but not CPIC guidelines. And for these, we find associations between warfarin and CYP2C19 and nicotine and CYP2B6. And then we also looked for novel associations where we know that there's an interaction, but there's been no evidence of a pharmacogenetic association. So this we find cyclosporin and 2C19 and rabeprazole and CYP2C9. And one of the ones I wanted to highlight here that I think is particularly interesting is warfarin and CYP2C9. You can barely see it, but there's this little dip down in normal metabolizers. And what this actually is an intronic variant in CYP2C9 that is extremely correlated with warfarin dose. So you can see that carriers of this allele, this variant, end up on a much lower dose than those who have the reference allele. It's unclear what this variant actually does, but it is listed in GTEx as being associated with expression of actually CYP2C9, but not in the liver. So it's unclear what effect this would have on warfarin dosing. The second analysis we did was the differential drug response. So as I said, this is just looking for a difference in side effects or other diagnoses in the year following the first exposure to a drug. So for this, we have every diagnosis that each patient had, and we're just going to look across all patients over this timeline to see whether there is an increased or a decreased amount of diagnoses from normal metabolizers or normal functioning individuals for each gene to anybody who's non-normal. And we get a couple of interesting things here. I'm only highlighting 2. There's a few more in the paper. But we get another sort of true positive confidence builder here, simvastatin and soft tissue disorder. So everything that falls under the soft tissue disorder umbrella is sort of like general pain, joint pain, muscle pain. And these are well-known side effects of statins. In fact, they're so well-known that the CPIC has written guidelines for SLCO1B1 phenotype and simvastatin-induced myopathy. This is a very severe condition, whereas muscle pain is sort of more benign, but we still see an increased incidence of muscle pain in simvastatin users. The second one I wanted to highlight was herpes zosters and citalopram. This one was very strange at first because why would citalopram be associated with shingles? But we actually see that intermediate metabolizers have about half the incidence of shingles diagnoses as normal metabolizers. which is bizarre, but there is a paper that describes increased resistance or immunity to the varicella zoster virus, which is the herpes zoster virus, in depressed individuals on antidepressants compared to depressed individuals not on antidepressants. So it's possible that there is some true association here where there's basically some protective effect with maybe having more of the drug circulating in your system for these intermediate metabolizers compared to normal metabolizers. And we're actively following up on these findings. I want to thank my lab, especially Russ and Adam, and the Pharmacogenomics team, Michelle and Katrin. They've been very helpful with these projects, all the participants in the UK Biobank, and feel free to reach out and ask any questions. Thank you.
Thank you very much, Greg. So we have time for a few questions. I can start with one. I was wondering what fraction of the associations you identified were due to coding versus non-coding variants?
So we map all the variants in genes to star alleles, and the vast majority of those are coding variants. There's some, like in CYP2C19, for example, has a eqtl upstream that is associated with being a rapid or ultra rapid metabolizer. But for the most part, they're coding variants.
Are you surprised by that, or is that what you expected?
No, that's about what I expected. Like all the previous work that has defined phenotypes for star alleles has found that most of the variants that are impactful are coding variants. But there very well could be eqtls that are important for function, but they just haven't been discovered before. But I only looked at known variants. This is things that we already knew about. So if there's anything unknown, and I didn't check it at all.
I see. Okay. We have time for other questions?
Yeah, I have another question. So I'm not super familiar with the UK Biobank dataset, but I suppose it's from UK, and there's a certain population there. So can that affect the study somehow? Or is it like
absolutely
very well?
Yeah. So this is I only did this among individuals of European descent. I think it's like 95% of people in the UK Biobank are of white British ancestry. So it's a very white population, but there are a few thousand, or some slightly more than that, individuals of non-European, like African and Middle Eastern and Indian, some Asian descent. But there's certain star alleles and phenotypes, like pharmacogenomic phenotypes, that are much more common in individuals from other populations. So, for example, like CYP3A5, most individuals of European descent don't have any functional copies of CYP3A5, but among African populations, having functional CYP3A5 is more common, and that could very strongly affect drug response. So it'd be important to look and see in other populations what we see as far as pharmacogenomic phenotypes and how that affects drug response.
Russ Altman asks, how are you following up on things like the intriguing herpes association with citalopram?
What a good question, Russ. We have a collaboration with Lisa Bastarash at Vanderbilt, where we're looking to follow up on some of these kind of intriguing hits that haven't really been reported for, to see if they find the same associations in their data. They have, I think it's like approximately 150,000 individuals that are genotyped, and we've been running PGX-POP, our pharmacogenomic assignment tool, on those individuals. And then Lisa's going to look into the phenotypes in their clinical records. But we don't have any results yet from that.
An anonymous attendee asks, is there any evidence of epistatic effects on drug metabolism?
Not that I'm aware of. I mean, maybe I don't understand the question exactly. But the way that I'm interpreting it, I guess, no.
Okay. Yeah, I mean, I would interpret it in terms of whether you see any kind of genetic interactions among any of the variants.
I mean, so for the gene that I'm most familiar with is CYP2D6, and there's been no evidence that, so like, I guess if you have a deleterious variant that poorly affects function or causes the protein to not function at all, there's no evidence that other variants will recover function. So I guess that would be it. Interaction, episodic effects. But as far as I know, there aren't many examples of that.
So Jan Sokol asks, when you mention the UK Biobank's phenotype data, are you referring to diagnoses recorded as ICD codes, or are you referring to other data, for example, responses to questionnaires?
For this analysis, I didn't do anything with the questionnaire data. It's entirely derived from the clinical records. So for the ICD codes specifically, there's the longitudinal data from the general practitioners where they get diagnoses over time from the GP. And then the questionnaire data is, for those unfamiliar with the UK Biobank, there's like an intake form where they get, they submit all these like lengthy surveys, and they may say like what diseases they have or don't have. But that's been shown to be less accurate than the ICD codings. And then since I was interested in diagnoses or like side effects, those sorts of things after going on a drug, I wanted that window of time after they've been exposed to the drug.
Okay. Carl Keat asks, for drugs with a lower proportion of genetic variance in response, are there strategies for generating a more precise prediction of drug response, potentially integrating other data?
Can you repeat that?
For drugs with a lower proportion of genetic variance in the response, are there strategies for generating a more precise prediction of drug response, potentially integrating other data?
I guess so. I understand that to be that if there's not a very strong genetic effect on drug response, I guess so. The thing that you could do that would really improve this kind of thing would be to incorporate lab values. So if you have measurements for like if you're trying to titrate the dose, then you can measure how much of the dose is circulating. That would be the best way to get at the actual pharmacogenomic response, because this is like just did they change their dose, which like for some drugs you may never change your dose even though there is a strong effect based on metabolizer status, or you might experience some side effects but you might not go to the doctor for it and report it to the doctor. So to better get at those things where it's sort of less obvious or the effects are less severe, that you would actually make an action based on it, then you would need something like that, laboratory measurements.
Okay.
Which there is some of
Marilyn
the UK Biobank, but I haven't looked at that.
Okay, thank you. So Marilyn Ritchie wants to go back to the earlier question about coding versus non-coding variants. She writes, a significant number of star alleles in CPIC guidelines are non-coding, not mostly coding. And so does PGX-POP only focus on the coding star alleles?
Yes. So, I mean, there absolutely are a lot of non-coding variants. And the data that we use for the study is the imputed data from genotype, which does include non-coding variants. So for PGX-POP, it'll call whatever is in the VCF that's input. So if it's exome data, then obviously it's going to miss the non-coding variants. But here we did use non-coding variants when available to the degree that they're accurately called.
Okay. Dana Crawford asks, the requirement of 5 prescriptions, was that just for the maintenance dose analysis? If also for the side effects analysis, is it possible you missed strong signals for patients who had 1 prescription and a serious or really annoying side effect?
Yes, that was only for the dose association. Just because if they only got 1 dose and went off of it, then we don't know how to define their like stable dosage over time. So, but yes, that question is exactly why we just did a single dose for the other association. If they went off of it and had some terrible reaction, then we want to know about that and have that in our analysis.
Okay. Joanne Bergot asks, are any of the drugs you studied prescribed for multiple conditions, and does this impact on the dose and or genotype dose relationship?
Yes. So many of the drugs are prescribed for multiple things, and that's definitely a limitation of the study. The best thing to do would be to kind of group by phenotype for indication that they were actually prescribed the drug for. But that's not something I did here. But that could certainly affect the dosage if there's a population of patients that have some indication that you need a higher dose for versus another indication that a lower dose for, that would affect the results.
We have an anonymous attendee question. What features did you include in your covariate linear models?
Age, sex, is age at the first prescription, sex, BMI, the first 4 PCs from principal component analysis of the genetic data, which should capture some ancestral effects. I think that's all included. That's certainly in the paper. Yeah.
All right, great. Thank you. We have 2 more questions in the Q&A. So Greg, if you could address those offline, that'd be great. And thank you again.
Yep, thank you.
And we have the last presentation now by Anmol Singh on incorporating of DNA methylation into eqtl mapping in African Americans.
My name is Anmol Singh, and today I will be presenting on the incorporation of DNA methylation into eqtl mapping in African Americans. DNA methylation plays an important role in the regulation of gene expression, which in turn affects many complex diseases and traits. Several investigators in the past have integrated genome-wide sequencing data and DNA methylation to uncover SNPs that significantly associate to CpG methylation, also known as methylation QTLs. These studies have found that methylation plays a significant role in the onset of diseases and phenotypes such as OCD, prostate cancer, and drug response. Most of these studies, however, have been conducted in populations of European ancestry. Minority representation in genomic studies has been very sparse, as shown from the figure on the slide, which leads to a lack of matched genomic and transcriptomic data in populations other than European descent populations. Africans in particular are the oldest population and thus have more genetic variation than other populations, which leads to disparities in diseases such as hepatocellular carcinoma, as well as other phenotypes such as drug response. Which leads to the significance of our study, which is the first investigation of the effect of DNA methylation on eqtl mapping in African American hepatocytes, which may help explain the role that DNA methylation plays in the health disparities observed in African Americans. We first started off by conducting whole genome-wide genotyping, DNA methylation analysis, and RNA-seq on our hepatocyte samples. We then performed standard QC protocols on all 53 of our samples. After this, we calculated the CpG sites within a 2.5 kb window of each SNP in our data set. And then we calculated methylation as a SNP-based covariate using the following procedure. If a SNP only had 1 CpG site linked to it, we calculated the weight of that CpG site as 1 minus the distance from the CpG site to the SNP over 2,500, where 2,500 indicates the maximum window size in our analysis. If we had a SNP with more than 1 CpG site linked to it, then we would calculate the initial weight using the equation listed above, but we would divide this initial weight over the sum of the initial weights of all the CpG sites linked to that SNP. After getting the final weight of each CpG site, we calculate the final SNP-based methylation value as the sum of the final weights times the methylation values for each CpG site linked to that SNP. Then, after calculating the SNP-based methylation value, we inputted our data into LA Matrix, which is an R package that has increased power and controls false positives when gene expression differs by a locus-specific covariate such as methylation. This program outputs the genotype gene expression associations within a cis region or 1 megabase around the gene. And we call these associations methylation-adjusted eqtls. After gathering our methylation-adjusted eqtls, we compare them to PC-adjusted eqtls, which were ran on the same data set, but were adjusted for global ancestry instead of methylation. Finally, we split our eqtls into 3 different groups, which were eqtls that were significant only with methylation adjustment, eqtls that were significant with PC adjustment but increased in significance with methylation adjustment, and eqtls that were significant with PC adjustment and decreased significance with methylation adjustment. We then compared these 3 groups to GWAS ontologies. For our PC-adjusted versus methylation-adjusted eqtls, we found that there was 308 eqtls that were significant with only methylation adjustment, 19,567 eqtls that were significant only with PC adjustment, as well as 1,954 that were significant with both. As shown in figure B, we found the negative log 10 p-values for PC-adjusted eqtls versus methylation-adjusted eqtls, with the color of the dots in the figure indicating which group those specific eqtls belong to in the methylation-adjusted eqtl dataset. We also ran an effect size analysis on all three of our methylation-adjusted eqtl groups, and all three groups showed a high correlation of effect size, and the Spearman correlation from this analysis is shown on the slide. For our GWAS associations, we found that a significant enrichment for digestive system disorders in the became significant group, no significant enrichments for the increased significance group, and significant enrichment for liver enzyme measurements and immune system disorders for the decreased significance group. We measured significance as FDR less than 0.05. For the became significance group, we found a key SNP gene pair between a SNP previously associated to primary biliary sclerosis and PKNP. And in the decreased significance group, we found two key SNP gene pairs between two SNPs and SORT1, which were previously associated to LDL cholesterol levels and lipoprotein measurements. Now, these two SNP gene pairs were still significant in our with methylation adjustment. However, the significance decreased considerably when compared to PC adjustment, indicating that methylation potentially plays a role in these SNP gene associations. We also ran a novel e-gene analysis for our methylation-adjusted eqtls, and we found 179 total e-genes through methylation-adjusted eqtl mapping at an FDR less than 0.05, as well as 80 e-genes that were not found significant with PC adjustment. Two key e-genes that were not found with PC adjustment were related to hepatocellular carcinoma, and they were GSTM3 and HSPA6. As shown from the figure on the slide, we can see that GSTM3 gene expression and DNA methylation have an inverse relationship, whereas HSPA6 gene expression and DNA methylation have a direct relationship. Also, there was 18 CPG sites that contributed to the GSTM3 association and 7 CPG sites that contributed to the HSPA6 association. Now, for our e-gene analysis, in previous studies, we found that high expression of HSPA6 and low expression of GSTM3 are related to hepatocellular carcinoma occurrence. We found an inverse relationship between GSTM3 and methylation in our study, as well as a direct relationship between HSPA6 and methylation in our study. Thus, our e-gene analysis results indicate that higher methylation levels could be a risk factor for hepatocellular carcinoma. Furthermore, the A allele of the SNP associated to HSPA6, which is also associated to higher HSPA6 expression and higher methylation levels, is not found at all in European ancestry populations, as shown from the pie charts taken from the Ensemble database at the top of the slide. For our GWAS associations, we found a key association between a SNP previously associated to primary biliary sclerosis and PKNP. This SNP in the primary biliary sclerosis study was associated to SPIB, and this association was assumed from the intergenic location of the SNP within the gene. Our study adds more weight to this finding since our study was done in hepatocytes and we used gene expression in our study. Furthermore, PKNP in a previous study was found to influence DNA repair after damage from oxidative stress. Thus, this SNP could be potentially involved in this process as well. Some limitations of our study were a small sample size, so our analysis was underpowered. We assessed methylation with an Illumina EPIC array, and thus we were limited to CPG sites on the array. Our results were only applicable to hepatocytes since that's the tissue that we use. We assumed that DNA methylation closer to a SNP causes a larger effect when that could not be the case. And finally, we assumed that the distance of 2.5 kb for our window size when CPG sites further away could have an effect on gene expression as well. In conclusion, this is the first study to explore the effect of DNA methylation in eqtl mapping in African Americans. The African American demographic is widely underrepresented in genetic studies, and their greater genetic diversity may allow us to find novel SNP gene pairs as well as population-specific SNPs. Thus, our findings can be used to understand how DNA methylation potentially plays a role in complex diseases and traits that affect African Americans. I would like to thank my PI, Dr. Manoli Pereira, as well as all of my co-authors for their help throughout this process. I would like to thank our funding source, and I would like to thank the Pacific Symposium on Biocomputing for giving me the opportunity to present today. Thank you.
Thank you very much, Anmol. Do we have you live? There you are. Maybe I can start with a question. I was wondering if you've looked how much methylation helps in analyses looking across different ethnicities. So you showed nicely how it improves your power for African Americans. How much does it help in other groups?
So the studies that I mentioned in the background were all conducted in European ancestry populations. So it definitely showed an increase in the findings there. I haven't really looked at any other populations though, besides that.
We have time for some additional questions. All right, well, thank you very much, Anmol.
All right, thank you.
Thank you very much, everyone. We are done with all the presentations. So now I believe we will move to another room for live discussions where everyone.
If you look in the chat, there's a link to the live discussion. So we'll all run over there, grab yourself a snack if you need it, and see you soon.
Thank you. Bye.